Hello, hello. Can everybody hear me? If you can hear me, type yes or something in the chat box. I just want to see if my audio is working well. I don't see any response. Okay, awesome. I see one yes. So, okay. Well, I hope everybody had fun at Online Town. And our next speaker is Dr. Okay, I see a lot of yes. Awesome. Okay, and our next speaker is Dr. Eilif Muller. And Dr. Muller obtained his PhD at University of Heidelberg in Germany and worked as a postdoc at EPFL in Switzerland before he joined the Blue Brain Project, where he led a team of researchers that developed, refined, and studied the well-known simulations of neocortical tissues. He's interested in understanding the principles of neocortical architecture, neural coding, and coding and plasticity. He's going to give us a talk on bridging scales of intelligence from biophysics and ConvNets. So I'm going to invite you on screen. Okay. Okay, he says he accepted. Awesome. Hello. I can't hear you. Have you muted your mic?
How's that?
Okay, perfect. All right, so take it away. I'm just going to give you the screen.
Okay.
Right. Sorry, Dr. Muller, I have to interrupt for a second. You sound, your sound is kind of on and off. It's, yeah, your voice is kind of breaking. Okay, so I think that's not just my internet. Okay, so let's wait for Dr. Muller to come back. Were you guys at online now? You guys have fun? Okay, I'm going to audio something. Okay, well, how about my sound? Yoga mentioned that my sound is not perfect. Can everybody hear me clear? Okay. Okay, so apparently now we can. And Dr. Muller is still working through the connection, I think.
now. Oh dear.
Okay.
I'm purple.
You look purple.
Oh, that's great. Okay.
You sound great now.
Can you send? I'm going to disconnect one more time. Can you send me a? I'm really sorry for this. Can you send me a link one more time? I'm going to disconnect again.
Absolutely, yes.
Actually, maybe let me just toggle the video on and off. There we go. Okay, forget it. That's good.
Okay. Would you like to share your screen?
Yes. Okay.
All right.
So should I start from the beginning? I guess you didn't get much of my sound.
Sure, yeah.
Okay. So, thanks very much, Don Yang, for the introduction. So I'm a computational neuroscientist, and my dream is to understand sensory perception and how it is learned. And increasingly, I'm feeling the need to take part and be involved in the artificial intelligence community. And so it's been super exciting to be a part of the Montreal Neuro AI community since May 2009. And a huge thanks to the organizers for putting on this event, especially to allow this great scientific exchange during these challenging times. For the last 8 years, I led the team of researchers at the Blue Brain project that built data-driven, large-scale simulations of neocortical tissue, generally of rodents like mouse and rat. In 2019, so just before I departed from the Blue Brain project to come to Montreal, we reconstructed and simulated the mouse isocortex, which is a major part of the neocortex. It's about 10 million neurons and 80 billion synapses, and we did it at cellular level resolution. So you can see all the dendrites and axons making up this structure. So the neocortex is the brain region specific to mammals, and perhaps the defining brain region of Homo sapiens. The connectivity between cortical regions, as well as the topography and the region innervation maps, were reconstructed in this milestone by jointly analyzing the recently released Allen Institute Mesoscale Connectome Atlas and the whole brain axon reconstructions from Genelia mouse light. And it was really made possible by recently developed capabilities of experimental neuroscience and the emergence of these large-scale collaborative data collection efforts. So I'm not going to talk much about that. That's, of course, a fresh result. But if you're interested in the details of how we built that circuit, I welcome you to go and look at this paper that we released in summer of last year. And interestingly, you can download connectomes and play with them yourselves. And this could be an interesting starting point, for example, for deep learning architectures. So the cortical architecture is a repeating motif. These networks of areas process and generate, respectively, speech, language, vision, audition, touch, motor planning, and many higher cognitive processes. And they're also preserved generally to a reasonable approximation across species. So if you go from rodents, which have a 2 millimeter thick isocortical surface, to carnivores, to primates, to humans, what you see is a gradual increase in thickness, but largely the 6-layered architecture and the composition of cells is preserved to a surprising degree. Another interesting aspect of the neocortex is that it's modular and hierarchical. So the modularity implies that if we're looking sort of at one half of the mouse cortical surface, that we can delineate specific patches of the brain that are responsible for specific, for example, sensory modalities. So we can see here these little blobs, these orange blobs, reflect the barrels, the whisker system of the rat or the mouse to process somatosensory information from their whisker system, a very important sense for the rat. It rivals almost the size of the visual system in the rat. For the hierarchical aspect, we mentioned that the neocortex is a 6-layered structure. The input rises and enters into layer 4, gets processed then by layers 2 and 3, pyramidal cells in layer 2, 3, who send an axon then up to feed into the layer 4 of a subsequent higher region in the cortical hierarchy, according to the textbook view. Of course, there are nuances that researchers are exploring in recent years. And also, there's a descending pathway. So layer 5, these very impressive layer 5 pyramidal cells are collecting input across the layers up into layer 1 and sending axons down to lower cortical layers and innervating into layer 4 and layer 5 and 6. So we have these 2 pathways of activity propagation, the ascending pathway and the descending pathway, and this defines the hierarchy of cortical processing. So it's an interesting historical fact to note, and I think relevant for this community, to point out that for those who are not aware, that ConvNets are actually a model, or were intended as a model of the ascending pathway of the neocortex. So the original convolutional network was the neocognitron developed by Fukushima in the 1980s, and his work was inspired by the mammalian visual system. So the work of Hubel and Wiesel into the cat visual system, and in particular, they identified this system in primary visual cortex and subsequent areas down the hierarchy of first simple cells receiving the input and learning to become orientation selective, and then complex cells which integrate over wider areas and introduce invariance. And so basically what we can see here is the inspiration for the commonly, so even modern ConvNets employ a similar architecture. So we have an input layer, multiple feature planes, computing features based on weight sharing across locations, where these shared weights can be learnable, and subsequently a blurring and subsampling operation to introduce invariances in the feature hierarchy. So in Fukushima's formulation, only synapses going from the simple cells to the complex cells were modifiable, and the cells going for, or the connections from complex cells going back into simple cells at the next higher up region were not modifiable. So I'd like to ask the question, are ConvNets a good model of the neocortex? And moreover, why should I care? So in particular, some of my colleagues may be surprised. I actually think that the neocognitron is a great starting point model. But if we're interested mostly in solving computer vision tasks, why do we care whether a particular model is representing the computational properties of the neocortex in particular? All we care about is that it solves the problem and achieves the task. So I would like to motivate why we should care about the neocortex by raising some of the key open challenges for ConvNets compared to the properties of the neocortex. So I would argue that the neocortex, learning in the neocortex appears to be very data efficient. It appears to have nice out of distribution generalization properties, and it appears to have nice properties of incremental or continual learning without catastrophic forgetting. Now, how these features come about in the neocortex still remains a mystery, but I think that what I would like to put forward in this talk is that by asking this question, relating ConvNets to the neocortex, that we may be able to get at new ideas for how to solve some of these hard problems. So I'd like to illustrate these properties of the neocortex with a quick little thought experiment. So let's consider, let's consider the example of a child learning a new novel animal. So I'm going to present you this new animal. I presume many of you have never seen this animal before. But I would argue that actually by seeing essentially one of these animals, that you have already achieved the task of knowing this is a jackalope, and you would be able to recognize a jackalope in subsequent frames. So where does this is apparently an n=1 data efficiency, in stark contrast to what it would take a ConvNet to learn this new novel animal class. So I would argue that this data efficiency comes from the child already having learned a good representation for the visual percept and the utterance. And that the learning of the jackalope is in fact an association or an associative operation of a particular subspace of the learned representation of the utterance with a particular subspace of the learned visual representation. And so, and this is very consistent with the view that's been put forward by Jan LeCun, that in asking the question, how do humans and animals learn so quickly? If intelligence or if learning is a cake, then the bulk of the cake is unsupervised learning, the icing on the cake is supervised learning, and the cherry on the cake is reinforcement learning. So interestingly, unlike modern variants, the original neocognitron learned its representations in an unsupervised way. Modern implementations of the neocognitron would still learn a readout in a supervised way by typical gradient descent on the features extracted on the head. But the hierarchy within the ConvNet and the training of the weights for the weight patches for each of the simple cell and feature planes within each of the layers is learned by competitive learning. And so here I'll explain, for those who aren't familiar, roughly how this works. So remember again, the complex cell layers are static, and they implement the invariance by blurring and the subsampling. For the feature learning, each simple cell plane learns to represent one cluster in the visual features that are presented to these feature planes. So again, there's weight sharing cross locations, and it's through competition between these feature planes that they repel and therefore span the space of the inputs. So a little bit more detail on how this works. For a given location, so if we're considering a cross-training patterns for a given training pattern, let's say that all of these feature planes are looking at this one location in the training pattern. Each of these feature planes can compute a certain activation given the input they're receiving. The maximum activated plane is the candidate to be representative for that specific location. And when it gets picked as the representative, there's a few more details that I'm brushing over. Then that particular location gets its weight patch reinforced with that particular input. And so what we can see after a few iterations of this competitive process on the MNIST data set is that before feature learning, if we look at the receptive fields, the weight patches of a few of these feature planes, they're all scrambled up, they're initially random, and that they learn to represent particularly oriented features in the visual scene. as one would expect of the simple cells in V1. And so in that sense, Fukushima really proposed this as a model of how, not only how the neocortex could learn to be orientation selective, but also the model that he proposed could do optical character recognition on something like the MNIST dataset that was available in the 1980s. So now competitive learning, competitive learning remains a distant caricature of the processes of neocortical learning, and those processes remain only partially understood. And it's actually for modern ConvNets in practical use, supervised learning has actually taken over and has been favored in practice. And so, however, there's a synergy between gaining a deeper understanding of the neuroscience of the neocortex and revisiting and refining its inspiration of ConvNets and their learning algorithms. So both the neocortex, studying the neocortex in and of itself, and convolutional networks such as the neocognitron, each represents progress at a different level of analysis of the neocortex as such in this 3 levels of analysis of understanding of intelligence proposed by David Marr. So David Marr said, if we want to understand something like the neocortex, we must understand it and analyze it at the level of the hardware implementation, that is, how is it realized in the biophysics of neurons and synapses. We have to relate that to how it represents inputs and outputs, and in terms of, and algorithms in terms of transformations. And finally, we have to represent it in abstract notions of the computational theory, the goal, and the strategy. And so for so largely, I would say much of the neocognitron is actually living in this space, and things like computational theory, goal, and strategy. This is very much the work of objective functions, of optimization, much as Konrad presented in the previous talk. And there's a nice review by Richards et al. That appeared last year on that topic. So Marr said that a neural net theory, unless it is closely tied to the known anatomy and physiology of some part of the brain and makes some unexpected and testable prediction, is of no value. Conversely, an appreciation for the intricacies and mechanisms at play at the biophysical level could inspire previously unexplored classes of categories of neural net theories, architectures, or learning paradigms, much like the work of Hubel and Wiesel for the original neocognitron. And I'm excited by the prospect that bridging these scales of understanding, it could reveal what's missing in ConvNets to solve these 3 open challenges and provide an expanded computational framework for making sense of neuroscience observations. So naturally, this remains a work in progress. As this is a student symposium for primarily non-neuroscientists, my task is to provide an overview of our integrated view of the neocortex that we received through the years working on these models at the Blue Brain project. And I wanted to give some hints of at some directions which I think merit further exploration towards solving these 3 challenges. So of course.
Hi. Wait.
Yeah.
Hi. Sorry, I just need to interrupt for a sec. Your slide is still on neocognitron and supervised learning. Is that supposed to be so?
No.
Okay, then I think there's a delay in switching the slides.
What do you see now?
I see your screen sharing, so I see your screen.
Okay, and what do you see now?
Still the same.
Screen sharing?
Screen sharing. So you're on the Chrome tab of the unique events.
That's not good. And now, do you see my presentation?
No.
Maybe we should reconnect?
Okay, sure. Let's try that.
Okay. I will stop sharing.
Yep.
And I will share screen again. Oops. How about now?
Okay, yeah. It's
my presentation? Okay, so you should see bridging levels of abstraction.
Correct.
Okay. So you didn't see the slide on David Marr's 3 levels?
No.
Okay, that's unfortunate. Technical issues. Apologies, everyone, for that. Can you see now David Marr's 3 levels of analysis?
Yeah.
Okay. In the interest of time, I think I'll plow ahead. I think it's going to be potentially a little bit confusing, but yeah, I think that's probably the best choice. So I was talking about David Marr's 3 levels at the hardware implementation, the representation algorithm, and the computational theory. And it's interesting to note that actually, as we have different subfields of neuroscience, computational neuroscience, and AI working at these different areas, predominantly neuroscience in the area of hardware implementation, computational neuroscience perhaps in the representation and algorithms together with AI. And AI, I would say, a very strong leader in terms of computational theory, the goal, and the strategy. So I'm now on bridging levels of abstraction. It's natural that these fields are actually working with different mathematical levels of abstraction for describing the underlying neurodynamics. So on the one hand, on the very far right, we have the artificial neuron, which is the fundamental unit of the deep learning field, which represents just a linear sum, weighted sum, plus an activation function. As we move to, probably the most common formalism in computational neuroscience is the point neuron, which, and finally, as we go to look at really the biophysical properties of these interacting systems, we can model at the level of biophysical neurons. And in fact, these areas can be connected through simplification from the biophysical side all the way to the artificial side. So for point neurons, what we're adding relative to an artificial neuron is the fact that neurons integrate and then fire or spike. That means that when a synaptic input arrives at a given time as an event now, in fact, time now is modeled generally as continuous in these cases, that a little exponential blip generally occurs on a state variable of the neuron. Usually it's the voltage. And if many of these synaptic events arrive within a certain amount of time, a threshold is reached and the neuron will emit a characterized event. So spikes are events, they're triggered at a threshold, and after a spike, they exhibit intrinsic dynamics such as refractoriness or other things. So biophysical neurons, in addition to this, also have a shape. So generally speaking, when we're experimenting on a neuron in a slice preparation, we actually have the ability to reconstruct, digitize the 3D shape of the neuron using a specially designed microscope. We can use these 3D shapes and apply a formalism developed by Wilfred Roll to model the electrical propagation of activity along these processes. And so, an important aspect of this propagation is that these synaptic disturbances that occur somewhere at some location in the dendrite, as they propagate, for example, towards the soma, which is where the spike generation zone of the neuron is located, that there is an attenuation and a spreading out of these synaptic events. So now, the nice thing about these biophysical formalism is that it's very closely related to the data that is accessible to a neuroscientist. That's partly why the Blue Brain Project started in the first place. Henry Markram is a world-leading electrophysiologist, which means he cuts up living, so he rapidly decapitates animals, cuts up slices that are kept, that remain alive in cerebral spinal fluid, and they have special preparations where what you see around, this is a microscope which has electrode systems surrounding the microscope, which allow us to approach near to one of the cell bodies, as we can see here, an electrode coming near to a cell body where we can attach to this cell, gain entrance into the cell, record and stimulate these cells. And it's through, and also by injecting a dye when we're undergoing these experiments, we can also reconstruct the shape. So using these techniques and by recording from many neurons simultaneously, we're able to gain a multitude of data sets that allow us to reconstruct in a digital form the circuits that are present, and thereby allow us to explore many experiments in silico or in simulation that wouldn't be possible in the experimental preparation alone. So the things we can collect, we can suck up the cytoplasm and assess the genetic material. We can assess protein profiles. We can inject standardized protocols to assess the firing properties of the cells. We can reconstruct the morphology, as I said, and also through paired recordings, we can stimulate one cell and assess the response postsynaptically of the activity of the innervating neuron on the postsynaptic voltage. We can also assess connectivity profiles by paired reconstructions of neurons. We can assess putative synaptic contacts within light microscopy, which we can then subsequently verify in electron microscopy. We can also use specially designed staining approaches to assess densities and proportions of different cells that we observe. So this was an effort that started in, well, the experimental data gathering has been going on for several decades in Henry Markram's wet lab and also many of his collaborators. And in 2005, the Blue Brain Project was launched to integrate all of this data, plus data that was available in the literature and produced by the community, to reconstruct at first just let's say a millimeter cubed of the neocortex. So what's some may say is the fundamental unit of neocortical tissue. And this was presented in this paper in Cell in 2015, where it provided basically the full detail of how this reconstruction, simulation, and how we, and very importantly, because this simulation is so complex, how did we validate to assess how did we assess the domain of validity of this simulation? And of course, validation doesn't mean that the model is right. A model is never right. A model can only be proved wrong. And so we use these validations to poke it and probe it and see under what conditions does it behave in the right way. It was released with two companion papers, one that made available the circuit to the community as a resource, and another paper that specifically went into how we predicted the connectivity between neurons in the circuit. So I don't really have time to go into the details of the whole reconstruction, but I want to try and fly through some of the aspects to give you a sense of what are the players in this neocortical circuit, and with the hopes that it might sort of expand your imagination with respect to what types of things we could do to build next generation convolutional networks, for example. So the first thing to observe is that the neurons, as classified by their morphological type, by their shape, can be classified into two broad camps. On the right, we have those that are labeled in red are the excitatory neurons. They make up 90% of the cell population, and with the exception of the spiny stellate neuron, they all are of pyramidal type, which means they have an integration zone locally around their spike generation zone, and they have an integration zone in the apical dendrite, and they're connected by nonlinear properties, as Konrad mentioned a little bit about in his talk. On the other hand, so now what's important to note about this population is that when they connect to another neuron, they can only excite the other neuron. So this is in contrast to modern ConvNets, where one neuron connecting to another neuron can either have a negative weight or a positive weight. This is strictly forbidden in the neocortex. Conversely, there's another class of neurons which are much more diverse and have rather exotic shapes that do the opposite thing. When they connect to another neuron, they pull the neuron away from threshold. That is, they inhibit the other neuron from spiking. And so we can go through some of the names of cells. So in broad strokes, we have the basket cells. These densely innervate locally around the neuron. They generally spike very, very rapidly in response to stimuli in a very sustained way, and are thought to be, for the first role, they're there to control feedback excitation. We have then other interesting cells that innervate across the layers. For example, Martinotti cells that seem to target the layer 1 tuft regions of the pyramidal cells. And this is an interesting mystery to further be elaborated on. There are a lot of theories, but as yet, the real functional role of these particular inhibitory cell types is still debated. We have also bipolar cells, double bouquet cells, and what's interesting is to note is that layer 1, like for the pyramidal cells, where they're sending their apical tuft up into the layer 1 space, there is a special population of interneurons that's living in layer 1. And it's also important to note that in layer 1, there are no excitatory cells. These neurons also exhibit an impressive electrical diversity, which means that when we stimulate them with a barrage of different stereotyped input patterns, we can classify them into broadly 10 or so different firing types. There's aspects of stochasticity, so regular firing or stuttering cells. There's aspects of, so we have delayed onset of firing in certain cell types. Another important feature is accommodation, that is that a cell through a fixed step stimulus will initially respond very, very rapidly and then decay its firing rate. And also some cells, as Konrad pointed out, will burst initially when they receive a step stimulus and then maintain a fixed firing rate. And then we have non-accommodating cells. As I mentioned, the basket cells are typically of this class. They can fire very, very rapidly at high sustained firing rates. So these diversity of firing types are made possible through a diversity of ion channels, specific pores that are selective to particular ions like sodium, potassium, and calcium embedded in the membrane of the neuron, and that are dependent on various aspects like voltage or concentrations of other ions in the cells. And it's the interacting dynamics of these ion channels that impart this bouquet of different firing types. And so, it may be seductive to think that an accommodating cell is just a cell that's getting tired, but in fact, this is not the case. There are specific mechanisms within the neuron that after a spike will will actively hyperpolarize the cell to prevent it from to slightly reduce its propensity to fire, and these accumulate over time with long time constants to result in this reduction in firing rate over time. And it's thought that these have important aspects of the temporal dynamics of coding in neurons. In particular, they select for changes in stimulus as opposed to coding in a linear way. Now we can use special staining techniques to assess the proportions of these different cells across the different layers. And we can use standard cell counting techniques across the different layers to assess their densities. And this allows us to place in a stochastic way locations within our circuit, after which we can place the morphologies and use a detection of touches plus pruning to reconstruct a null hypothesis as to the connectivity of the circuit. Now, of course, this is a scrambled egg. The circuit has not yet learned anything, and there's important higher order connectivity that you will get from learning processes that go on the circuit, and I'll talk about that in a minute. But this connectivity, I want to stress, is heavily validated, and it's consistent with available data with respect to things like Bouton densities along the axon, the number of synapses mediating a particular connection, and other interesting statistics. So this is one point where we really did extensive validation on the circuit. Next, we can reconstruct the dynamics of these neuron models according to the experimental data that we have for the specific firing types. We have we can take morphologies, and through standard simulation tools, we can use genetic algorithms to optimize the parameters of these simulated in silico neurons to match their experimental counterparts. And we generally use genetic algorithms and a multi-objective optimization approach. Lastly, we have to reconstruct the synapse dynamics. And Konrad talked a little bit about this. So generally speaking, one of the leading models of synaptic transmission is that it exhibits dynamics and that it is stochastic. And so the way we model this is that at the presynaptic terminal, you have a population of vesicles that are readily releasable. These, and you have a postsynaptic neuron that has receptors that will result in voltage transients postsynaptically when one of those vesicles succeeds at being released across the synaptic cleft. When an action potential travels down the axon of the presynaptic neuron, these vesicles will release with a random probability, which has its own dynamics. So the probability of release can alter with can be altered on short time scales with the history of the neuron on the time scale of seconds. But also, when synapse is released, there's a dynamics of replenishment of these readily releasable sites, and this the time scale of recovery results also in depression. So this means that the readily releasable pool can be depleted over time scales of seconds for many pulses, resulting in a rapid attenuation of the synaptic response postsynaptically. What I also want to stress here is that these blue traces of a bunch of subsequent pulses, in this case 8 or 10, while they exhibit depression, you can see for each of the individual traces in gray, this blue is an average of about 30 trials. Each individual trajectory is highly stochastic, and there can be, depending on the amount of vesicles populating the readily releasable pool, you have a high if it's depleted, you have a high probability of failure, which makes synapses incredibly a large source of noise in the system. So once we've put all these things together, we can we've developed the technology to simulate these networks on supercomputers. And so here you're seeing a network of the 6-layered cortical structure with 217,000 neurons. And now we're seeing, we're visualizing about 1% of the neurons, and we can see what's happening here. You can see on the left the time course of the activity in the network in response to a thalamocortical stimulation. So let's say input into the network. So what you can see is that as what you're seeing in terms of the color of the neurons is the voltage of the neuron. When a neuron flashes white, that means it's firing. And so what you can see is that when a stimulus enters the network, a subset of the neurons are activated, and that this activation propagates through the network across the layers in interesting and beautiful ways. So the time scale of the simulation you can see is on the order of a few seconds. So this is a dynamic event of a flick of a whisker, so to speak, a model of a whisker flick, and that input propagating into this network. So I mentioned also that the synapses within this network should learn or change. And so typically this is modeled in biophysical networks by something called plasticity, which means that the synaptic weights and also the connectivity itself can be modified as a function of the activity in the network. So for example, the weight could get stronger or weaker, and this can be expressed in a time scale of minutes, or a synapse can be added or retracted on the time course of an hour. And this synaptic plasticity is broadly thought to be a biological substrate for learning and memory. So I'd like to announce a paper that recently came out. This is a long body of work by a PhD student that I co-supervised at the Blue Brain project to develop in collaboration with Michael Graupner at CNRS in Paris, based on his previous work on calcium-based modeling, which we chose because of its biophysical relevance with respect to a lot of the synaptic transmitters that we already have present in the dendrites, such as NMDA. So all of these synaptic transmitters that are present on the dendrite of the neuron, they result in calcium influx into the postsynaptic dendritic compartments. And it's this calcium that is thought to be the driver of plastic changes in neurons. And so generalizing Michael Graupner's previously published model to work within the biophysical framework of our neurons and our synapses, we were able to find that we could parameterize this model in a way that a single model and a single parameterization could account for the available data on neocortical plasticity across, from neuron types across layers, but also for different spatial distributions, where it's known that complex flips and changes in the outcome of plasticity experiments can occur. And this was achieved by actually using our network as a sort of in silico experiment and reproducing as many of these experiments in the literature, and then running parameter optimization techniques en masse to
interrupt for a sec. It seems that your slide is still frozen on the visualization of the neurons.
Oh, really?
Yeah.
On this movie?
Oh, yeah. Okay. I think it's better now. I just want to make sure that you're using the correct slide.
You should see plasticity now.
I'm seeing your Chrome window. So if you want to just share it again.
Yeah, I'm going to share screen. Sorry for that. Okay, you should see plasticity now.
Yes. And also we can just go a little bit over time, maybe like 10 more minutes, if you have anything to wrap up.
Okay.
I have a couple questions.
Okay. Sounds good. So I guess I'll plow ahead in the interest of time. The other aspect that I'd like to point out is that this biophysical network now actually allows us to perform a principled study of prominent simplifying assumptions in use in the computational neuroscience community in transitioning to point neuron models. And this was work by Christian Rossert, and collaborators. And basically how this works is we found a way to compute for each synapse a compensatory filter that allows us to displace synapses to the soma and thereby convert the dynamics of the propagation in dendrites to something that's consistent with a point neuron network. And we can show that by comparing the green, which is this approach, and the blue, which is the reference, compared to an assumption which just displaces the synapse, which is the red, we can see that the approximation is doing a great job at reproducing the voltage trajectories in blue and in green and red of these neurons under an in vivo-like barrage of inputs to their dendrites. So we use this to demonstrate that, in fact, a common assumption to neglect the dynamics of synapses, depletion and facilitation, and also to remove these accommodating aspects of pyramidal cells and, for example, Martinotti cells in the network, dramatically alters the dynamics of the network. So if we take our best guess at a point neuron network, which is the black, so if we look here down below on the right, we can see dynamic responses in terms of spikes per unit time for a whisker flick. We can see that the reference network in blue and our simplification approach in black, they're doing a pretty good job at agreeing with respect to stimulus response for specific neurons in the network. What we find is when we neglect these dynamical properties in the network, that many of the neurons in response to these patterns don't respond at all. And this is, in fact, not surprising. These dynamics of the neuron are known to amplify transient responses to stimulus changes. So this, I think, demonstrates that we have to be careful in making ad hoc simplifications to link biological mechanisms to function, because these are very complex systems. We can summarize using similar techniques, which are available. So the mathematics for simplifying point neurons to mean field equations is quite well developed. In fact, we can get a sense of, in fact, what we're losing as we're transitioning from biophysics over to artificial neurons. And I think one of my message here is that I think that it's an exciting direction to consider, from the very far left to the very far right, what is being lost until there, and perhaps devising new formalisms on the artificial neuron that can actually, that we can re-inject some of the key aspects of the biophysical neuron into those frameworks. And so I wanted to give just quickly in the last 5 minutes or so, if I may, some of the insights that I've been thinking about from the biophysical universe that I previously worked in terms of shifts in perspective. So one of the things that I feel is really important to drive home is that connections between neurons are actually multisynaptic. So when a pyramidal cell connects to another pyramidal cell, it doesn't make one synapse. It makes many, many synapses spread over the dendritic tree. That the synapses mediating to the dynamics of the synapse in terms of learning is actually on the long time scale considered by many to be bistable. That is that when something makes a decision to learn and to consolidate, that consolidation is that strong consolidated state is very long lived. Moreover, that networks are consistently rewiring, but that consolidated synapses will persist over very, very long durations. And I think this is an interesting to put this in perspective of recently developed a recent paper that's used iterative pruning to pack multiple tasks into a single network. And I think this is sort of, it feels like it's going in the direction of structural plasticity, but of course this is done in a very isolated way for each task. And I think there's promise here to try and find a way inspired by the biophysics to try to do this in an online way as a solution to continual learning. Another thing, and that's something that I'm very interested in pursuing discussions with people in Montreal on those topics. Another interesting observation is that shape appears to matter with respect to coding on dendrites. So one of my favorite papers from 2016 was the observation using light techniques that the orientation selectivity of synapses along dendritic branches actually formed functional clusters. That is that you could find groups of synapses along the dendrite that shared the same orientation selectivity. And one explanation for this is that functional clusters cooperate to produce NMDA spikes, and NMDA spikes trigger calcium. This image being true is a hotly debated question, is whether cortical neurons can respond reliably at all, given the immense noise and chaos that they face in the recurrent network. And this is something that we investigated with Max Nolte, who was a PhD student in our group. And basically, we used a restarting setting to assess the chaos in the network, which indeed the network, a checkpoint of the network restarted with different random number seeds. These two equally valid trajectories from the initial state diverge incredibly rapidly within a time scale of 100 milliseconds. But what we found is that when we provided input to the network that was consistent with the tickle that would be coming from the whisker system of the rat, that this could evoke very reliable firing in a subset of neurons in the network, which is consistent with this view that clusters could actually be present on dendrites to evoke these NMDA spikes. And lastly, we found that in this network, unlike a random network, it's highly overexpressed in terms of all-to-all connected cliques of neurons, where we can count the dimension of the clique as the number of neurons involved in this all-to-all connection. And what we found is that we found many with surprisingly high degrees of all-to-all connectivity. And these subnetworks, when analyzed conditionally, have a much higher mean correlation. And they seem to be driving correlated spiking within the network, which could be a driver of plasticity. I'd like to do a shout out to some recent data done by the Orion Lab at UDM that's investigated, that's used light-based two-photon imaging and stimulation techniques to actually assess that indeed clustered synapses that are stimulated nearby to each other can totally abolish the depression window of plasticity protocols and result in only a potentiation window, which is an interesting observation that synapses can co-potentiate, and it's, in a sense, it's unsupervised. It has an absence of third factors. Now, I just lastly want to say that I've totally neglected in all of this view the role of descending pathway and bipolar integration of PCs in learning. I think this is also super, super important. And I'm very excited to be here in Montreal with people like Blake Richards and Richard Noe in Ottawa to be able to look deeper into the interaction between these sort of ideas that we're seeing it with respect to mechanisms of unsupervised learning in the ascending pathway, and to marry that with, I think, very important ideas regarding how the ascending learning in the ascending pathway could be gated or interact with the descending pathway. So I think that it's important to put a caveat that there is no free lunch. I think that so unsupervised learning in the neocortex must rely on structure at a specific spatial and temporal scale. The sensory organs, the inputs, have evolved to give neocortex what it needs to undertake this task. And I think the neocortical, so we could state this as the neocortical solution requires a neocortical problem. This is what Blake and colleagues in the review called the brain set. And I think that if we want to look for unsupervised learning, we really need to make sure that we get the structure of the input calibrated to the elements that we're employing. For example, a fly neuron has a much faster time scale and will learn different things than, for example, a sloth neuron. So these intrinsic timescales are indeed embedded in these neurons. So I'd like to close to say that I'm very excited by the state of the field. I think on the one hand, we have amazing new data sets that have just come out or on the cusp of coming out. We have this explosion in the field of AI, and I'm captivated by this idea that the convolutional network is in a sense a Rosetta Stone that allows these two fields to really communicate together when it comes to the idea of the neocortex, which is the brain structure I'm interested in. And I'm very much looking forward to a fruitful exchange between these ideas to, on the one hand, try to use neuroinspiration to solve some of the hard problems in AI, but also that the resulting computational and theoretical frameworks will elucidate some of the mysteries that we have in interpreting the data at the level of the neurons and the synapses. With that, I should acknowledge this is a huge team effort of all of the blue brain simulations that you saw is a huge team effort based in EPFL in Switzerland at the Blue Brain Project. We benefited from grants on supercomputers from Argonne National Labs and funding from the European Commission, and our supercomputer was housed in the Swiss National Supercomputing Center. And also a lot of this thinking with respect to how this relates to artificial intelligence has been done in the last year at Element AI. Thanks very much for your attention, and sorry, I hope I didn't go too much over time, and be glad to take some questions if there's time.
Thank you. And so sorry for going a bit over time, everyone. And because of our time limit, I'm just going to tackle the top question, the most upvoted question. I'm just going to read it to you. So the question says, how much should we rely on artificial deep neural networks black box in explaining the complex mechanisms that could be happening in the brain on abstract level as biophysical model?
Sorry, how can I see the question?
So it's in Ask a Question tab right at the bottom of the screen.
Okay, how much should we rely on artificial deep neural networks black box in explaining the complex mechanism that could be happening in the brain on the abstract level as biophysical model? So I think what you're saying is, if I understand the question, you're asking how is an artificial deep neural network a good model of the neocortex, really? And I think in the context of David Marr, I think, when we're analyzing the neocortex at the multiple levels, what the neocortex or what the convolutional network or the neocognitron offers is a computational framework to understand the neurons and the biophysics. Is it a good model? Well, models are never right. You can only prove a model wrong by testing it in a certain situation. So, of course, the model will be wrong under certain scenarios. And I think the exciting thing to do is not to claim that a model is good enough and we're done and we can go home, but it's really this. I certainly don't believe that the neocognitron is right. There's all sorts of aspects that are, that we could have elaborated on if we had more time, where it really is a vast oversimplification. Just to name the intrinsic property, when I gave you the example of simplifying from the detailed network to the point neuron network, already we could see that dynamics was a fundamentally important aspect of the network. Convolutional networks don't even have dynamics. It's a feed-forward path. The human visual, or the mammalian visual system performs saccades. There's interesting successions of visual frames from one to another. This is totally absent in the ConvNet. I think it's a super important fact for understanding unsupervised learning, for example. There's a lot you can learn from just, for example, I imagine you could learn aspects of segmentation just by succession of frames and co-movement of objects. And that's, you know, a problem that I worked on in the framework of ConvNets at Element AI. So I think the answer is absolutely not. I don't think we can sort of be happy with the ConvNet as a model, but I think, you know, when it's there to be shot down, and whenever something better comes along, then let's welcome it with open arms. But I think for now it does offer a useful abstract computational framework within to sort of arrange all of these gory, I think oftentimes they can feel myopic details of the neocortex. So it is important to, as David Marr suggests, I feel, analyze at these different levels and build bridges and relationships between them.
That sounds good. Well, I think that concludes our morning talks. Thank you so much, Dr. Muller.
Thank you very much for having me.
All right. Well, so our next session will be at 1 p.m. And that will be our lightning talks from our students. So for now, I'm just going to end this session, and we'll have a lunch break. And I'll see you guys at 1. Does that sound good? Right.